my name is Michaela, and today I'll be teaching about the processes of mitosis and meiosis. First up, we have mitosis. Before mitosis comes the step called the interphase. As you can see, the nuclear envelope and nucleus are still present at this point, and within this nuclear envelope there is chromatin. Due to the chromatin, the chromosomes are undiscernible at this point. In the corner, you can see that both centrioles are located in one pole, forming a centrosome. The first phase of mitosis is the prophase, and we would identify this as early prophase. As we can see, the centrioles are starting to navigate to their opposite poles, and spindle fibers are beginning to form. In this stage, we'll see the nuclear envelope and nucleus disappear, and the chromosomes will shorten and thicken to become more discernible. Here we see the cell in prophase. The centrioles are continuing to move to their opposite poles, and the nuclear envelope is continuing to disappear. We see that the chromatin has turned into the shortened and thickened chromosomes, which are double-stranded and held together by centromeres. At this point, we can see that the nuclear envelope has completely disappeared, and the centrioles, also known as asters, have almost made it to their opposite poles. At this point, we would consider it late prophase, and we'll see the spindle fibers start to attach themselves to the chromosomes to help push and pull them around the cell. Here we can see that the spindle fibers have attached themselves to all of the chromosomes and will begin to pull them towards the middle, leading into what we know as the metaphase. Here we see the metaphase. The spindle fibers have pulled all of the chromosomes into the middle of the cell, and they have lined up randomly and single file along the equilateral plate. There is no rhyme or rhythm to how these cells line up. It is completely random. It is also important to note that the direction of the chromosomes is perpendicular to the direction of the poles. Once the metaphase is complete, we see the anaphase occur. This is the phase in which the spindle fibers will pull on each identical sister chromatid to separate them and pull them towards opposite poles. This is the first step in creating two new cells out of the one original parent cell. Here we see the spindle fibers continuing to pull the chromosomes apart, helping us to form our two somatic daughter cells. We see a cleavage furrow beginning to occur, which is the first step in leading us towards our telophase. What we just identified would be considered early telophase, and what we see here now is telophase. The cleavage furrow is continuing to occur, helping us to create two new somatic daughter cells. The nuclear envelope is forming in both cells, and we see the chromosomes lengthening and thinning out. As you can see, the spindle fibers have completely disappeared. After the telophase comes cytokinesis, which is the final cell division. We see now that we have two brand new somatic daughter cells, and they have returned to the interphase stage. They will now be able to continue the cell cycle and repeat mitosis once again. Now that we have discussed mitosis, we are going to move on to meiosis. Meiosis is the process by which sex cells are formed. Instead of dealing with single chromosomes, we are going to be dealing with homologous pairs. First off, we are going to start with meiosis 1. Similar to mitosis, we see that meiosis 1 begins with an interphase. We see the nuclear envelope and nucleus are present, and within is the chromatin. The centrioles are located in one pole and have yet to navigate to their opposite poles. Here we see prophase 1. The centrioles are beginning to move to their opposite poles and spindle fibers are forming. During prophase 1, an important process occurs known as synapsis. This is when the homologous chromosomes line up to form tetrads. This is when there are two chromosomes, four chromatids, and it results in one homologous pair. Where the chromatids are overlapping, it is called a chiasmata, and it allows for crossing over of genetic information between the chromosomes. This correlates to greater diversity of the offspring. Nearing the end of prophase 1, we see the spindle fibers connecting with the homologous pairs, preparing for metaphase 1. Here we see metaphase 1 occurring. The spindle fibers have pushed and pulled the homologous pairs into the center of the cell to line up across the equatorial plate. The homologous pairs line up by random assortment, as the maternal and paternal DNA have no set association with the poles. This is important in creating genetic diversity among the offspring. Up next, we see anaphase 1 occurring. Similar to mitosis, we see the chromosomes splitting apart. The homologous chromosomes will separate, however the sister chromosomes remain attached. Two chromatids move towards each pole in opposite directions. This is known as segregation. This splitting is due to the action of the spindle fibers. Here we see the cleavage furrow occurring, which will help us to form our two new haploid sex cells. During the telophase, the cleavage furrow will occur and two new cells will be formed. Telophase 1 is where we will begin to see our two new haploid cells forming and how the original DNA has been split. As we can see in telophase 1, the nuclear envelope and nucleolus have returned, and we have two new haploid cells based on the original chromosomes and DNA we had. Cytokinesis has occurred, meaning the cytoplasm has divided to form our two haploid cells. Finally, we are going to discuss meiosis 2. In meiosis 2, the only difference is that there is no interphase as it goes directly from meiosis 1 to meiosis 2. So let's get started. Just to be clear, I only drew one of the two cells that was created in meiosis 1 just for ease of explanation, so we will only see half of the genetic DNA that was created, but at the end I will show all four cells that are created. Here we see prophase 2 occurring, where the centrioles are moving to their opposite poles, and the nuclear envelope will begin to disappear. Here we see late prophase occurring, where the spindle fibers have attached themselves to the chromosomes to start pulling them towards the middle for metaphase 2. Here we see metaphase 2 occurring. It is very similar to metaphase in mitosis, as the chromosomes line up single file along the equatorial plate by random assortment. After metaphase 2, we see anaphase 2 occur. The difference between anaphase 1 and anaphase 2 is that in anaphase 1, the homologous pairs are separating, and in anaphase 2, the sister chromatids are separating. We see the spindle fibers pulling the sister chromatids towards the opposite poles. After anaphase 2, we move into telophase 2. Here we see a cleavage furrow begin to develop, and the different strands of DNA become enclosed in a nuclear envelope. At this point, cytokinesis will occur, which will divide our gamete cell into two new haploid cells. At this point, telophase 2 would be complete, and cytokinesis would have helped to completely separate our haploid cells. If I had drawn both of the cells from meiosis 1, then this is about what we would have ended up with. Since meiosis is the process of gametogenesis, a male going through spermatogenesis would create 4 sperm, and a woman going through oogenesis would create 1 ova and 2 polar bodies. That is everything I have to share about mitosis and meiosis. I hope you learned a lot, and thank you for watching.